So before we can begin to dive into the process of meiosis, a very important phase will always happen before a cell division. It is not part of meiosis. It happens once at the beginning of the whole process of meiosis, just like with mitosis. And the very important thing to know that happens in interphase is the DNA replicates. So we did that in the previous section. But what that means is that these chromosomes are naturally single-stranded. But what happens from DNA replication is that this chromosome makes an identical copy of itself. And now they remain joined together by a centromere, which is in the middle of the chromosome. And you end up having, on the left-hand side of the arrow, you have two single-stranded chromosomes, which we call a homologous pair. And when they replicate their DNA, you end up with two double-stranded chromosomes. So you don't change the chromosome number there. You are simply changing the number of chromatids. So this is a centromere in the middle of the chromosome. That would be a centromere. So there are two centromeres on the left, so therefore two chromosomes, and two centromeres on the right, therefore two chromosomes still. But each wing of this chromosome is called a chromatid. So I call meiosis the section with a lot of C words. So far we've got chromosomes, we've got centromere. So this whole structure is a chromosome, that whole structure is a chromosome, but it's just double-stranded. And those two chromatids are held together by this black dot called a centromere. So we look at these as being a homologous pair because they're equal in size, like I mentioned in the previous video. You get one from your mom, one from your dad, and both of those chromosomes will replicate their own DNA to make two double-stranded chromosomes. It's still two because you count the centromeres. So let's dive into meiosis. So meiosis 1 has a prophase I, metaphase I, anaphase I, telophase I. You can show that the 1 is a simple digit or a Roman numeral. But like I said earlier, that the cells go from being diploid to haploid because homologous pairs are separated. So diploid means you have two sets of chromosomes. Haploid means you have one set. That's why we have an N for haploid, a single set of chromosomes, and a 2N for diploid because it's two sets of chromosomes. Now just remember, you get those sets of chromosomes as a result of a woman's egg cell being fertilized by a man's sperm cell. So those two cells each have 23 chromosomes. That is a haploid human cell, that is a haploid human cell. Now what happens is when that sperm cell with its haploid set, 23 chromosomes, fertilizes the egg cell that also has 23 chromosomes, you end up making an embryo or a zygote that has 46 chromosomes. So this is a haploid cell produced through spermatogenesis, meiosis. This is a haploid egg cell produced through oogenesis, also meiosis, and you end up getting a diploid zygote or a diploid embryo. Now, every animal and every plant has its own chromosome number. 23 is the human haploid number, 46 is the human diploid number. And this 46 is as a result of having 23 pairs. So you get 23 individual chromosomes from your dad's sperm cell, 23 individual chromosomes from your mom's egg cell, and those 23 chromosomes 1 to 23 join together to make 46, which is 23 pairs. So let's dive into prophase I. Prophase I has some very busy steps that are happening, so we need to remember that this is the busiest phase that occurs during meiosis in the entire process. The first step that happens is the nuclear membrane and the nucleolus disintegrate. We show this with a dotted line around the chromosomes and a pale colored sphere which shows the nucleolus disintegrating. That is the nuclear membrane disintegrating. We can also say disappearing. Then the chromatin network will condense. Normally chromosomes aren't visible like this. They only become visible like this X shape after DNA replication has occurred. And then the chromatin network is this mess of hair that you would take out of a hairbrush. And it needs to condense and come closer and closer together to form these distinct or clear individual chromosomes. So that would be the next thing particular to prophase I. Now, a reminder that these chromosomes are visible as two identical chromatids joined together by a centromere in the middle of the chromosome. The next thing is the centrosome splits. This whole structure combined is a centrosome, but one of these little rectangles is a centriole. Again, lots of new C words coming in here. So these centrioles split and actually start to form the spindle fiber, which is not visible in this image either, but other pictures will show the spindle fibers starting to form between them. Then, something that is very important in meiosis and prophase I particularly is that the chromosomes start to arrange themselves in what we call homologous pairs. So you'll have chromosome 1 from your mother coming together with chromosome 1 that you got from your father. Your father's sperm cell had that one. Your mother's egg cell had that one. They come in line next to one another. And they are homologous because they are the same size and they contain the same genes. But they lie next to each other and they overlap in a process called crossing over. So there's another C word. So this crossing over leads to the exchange of genetic material between your maternal chromosome and your paternal chromosome. And it will only happen with homologous pairs. So this chromosome here, number 3, is crossing over only at that one point. I will do a video on crossing over in a separate video as this one is already far too long. So let's hurry along to metaphase. So metaphase I is quite similar to in mitosis, where you see the chromosomes lined up at the middle of the cell. So metaphase for middle of the cell. And the other word for this is that they are at the equator of the cell. So now the spindle threads are fully formed between the two centrioles, and you will see these chromosomes arrange themselves in homologous pairs at the equator of the cell. So you've got a chromosome there and a chromosome below. They are homologous pairs, they are the same size. A chromosome there and a chromosome below, chromosome there, chromosome below. They can also be shown as this diagram being inverted by 90 degrees, and then they will all look next to each other. But the point is they are paired chromosomes. That is a very important distinction to make. All these chromosomes show crossing over where the genes have been switched or exchanged between the maternal and paternal chromosome. And what we say here, crossing over introduces variation. You can see that these chromatids are slightly different to each other on that chromosome and on this one. But another process that introduces genetic variation in meiosis is this process called random arrangement of chromosomes. So we say chromosomes arrange themselves randomly. And all that is that it is flux if the maternal one is there and the paternal one is there. If they are switched around, it will give you an entirely different gamete that is produced. So it doesn't matter which way around they are, it's completely random and it will affect the gamete that is produced. It could even make, instead of someone being born a boy, biologically speaking, it could make someone be born a girl if they got an X chromosome instead of a Y from their father. So you see these chromosomes lined up in homologous pairs and they're attached to the spindle by their centromeres. So these are the spindle fibers being shown with a dotted line, and they are all attached to the spindle by their centromeres. So now anaphase I takes place, where the spindle threads will contract, or the spindle fibers, you can use those terms interchangeably. They contract and they pull apart whole chromosomes to the opposite poles. So there was one member of chromosome 1 going to that top pole. There's the other member of chromosome 1 going to the bottom pole. So still in this picture, there are 6 chromosomes. You're always counting the centromeres. But actually you can see that if we were to draw a dotted line across the middle, if we were to complete the process of meiosis and we were asked how many chromosomes will be present in each cell at the end, you can see that there will be 1, 2, 3 in each cell. Because there's chromosome 1, there's chromosome 2, there's chromosome number 3. So what happens during anaphase I, very specific to anaphase I, not anaphase II, is that the homologous pair separates and one member of each pole is pulled to the opposite pole. So it's separating the pair. Now, anaphase can also be shown with the chromosomes being slightly V-shaped. That doesn't matter. They're still being separated, being pulled apart by the spindle. These are just shown as being a bit more flat or two-dimensional. Anaphase I is followed by telophase I, and what you see is you have 2 cells being formed. Sometimes they will show them as still being slightly connected with just a cleavage furrow forming between them due to cytokinesis. But you have these 2 groups of chromosomes that form at each pole. 1, 2, 3 chromosomes, 1, 2, 3. And these chromosomes are still double-stranded, which is really important for identifying telophase I. In telophase I, the chromosomes are double-stranded. In telophase II, you'll see it in the next video, the chromosomes are single-stranded. Now, each nucleus that forms has half the chromosome number as the original parent cell. The parent cell was diploid. In this picture or example, it had 6 chromosomes. Now we have only 3. So we have already halved the chromosome number. Then the cytoplasm divides through cytokinesis, and it's important to note that these two cells are both genetically different from each other. For example, to simplify it, this top cell has 2 maternal chromosomes and 1 paternal, whereas this one has 2 paternal chromosomes and 1 maternal. Now, these chromosomes will contain different genes if it was a paternal one or a maternal one. I apologize for this sincerely long video, and the next one will hopefully be a bit shorter.